So today I'm going to be sharing a recent paper titled Transcriptional and Epigenetic Decoding of the Microglial Aging Process, and came out just last week or so. Now, the overview is that age-related microglial changes increase susceptibility to brain dysfunction, and the study is mapping the transcriptional and epigenetic microglial changes in aging mice from 3 to 24 months. They find some sex differences and age-dependent microglial genes that change from across the age spectrum, and then looked in further detail at single-cell data and established a accelerated microglial turnover model, which they kind of present as a model of microglial aging, but it really is just rapid turnover. We'll talk about that more at the end. It's a lot of data, so I'm going to try to move relatively quickly through this. The first set was Bulk RNA-seq fact-sorted microglia from C57 mice, and they did many time points from 3 to 24 months. Not exactly the same time points in males and females, which is kind of interesting, and they didn't really explain why, but did say they didn't think it was a problem. But the key thing that came out of this is that across the spectrum from 3 months to 24 months, they see in the females there's a kind of distinct gradual changing of aging of the microglial expression patterns, where you see young, middle-aged, and old having these different patterns, whereas in the male mice, you don't see that. They saw a much more flip from a young phenotype to an old phenotype in the males. So that prompted them then to kind of look through what else is changing in these, and can we learn anything from that progression of the aging microglia. They did PCA of both of these and found distinct patterns of the young versus the old in these groups, in the males, and then young, middle-aged, and old in the females. And looking at the expression of specific markers within these, there are several of them, a few examples here, that correlate with age. So as the mice get older, the expression changes of these. They then identified specific genes that changed with the aging process in both females and males, and there are a set of significantly positively correlated age-dependent microglial genes and negatively age-dependent microglial genes. So these ones go down with age, these ones go up with age, and then a different set in males. There's some overlap in these, but not complete overlap in these. Interesting things to dig in for future analyses there. And when you look at the biological processes, of the top 10 significantly enriched genes, they point to leukocytes and chemokines, immune response kind of things, which may be not surprising for microglia. And then similar analysis with IPA, ingenuity's pathway analysis, and see kind of similar things there. We then looked at the overlap of these newly identified age-dependent microglial genes and damage or disease-associated genes, and there is some overlap there listed below. And then on the far right panel, also with microglial activation-related genes, and there's much less overlap there. So the kind of the takeaway is that the aging phenotype is not just equivalent to cell reactivity. It's not that you're getting hyperactivation of microglia with age. There's just kind of a different phenotype that is emerging. And here looking at the correlation of the disease-associated genes with the age-associated genes. So the next set, for the rest of the study, they looked specifically just at females and at 3 time points of young, middle, and aged, and dove into single-cell transcriptomics here and looking at, and you see the clustering in these t-SNE plots of distinct groupings of the single-cell profiles, and they're clustering for the young, middle, and old phenotypes. And then they did a further unsupervised classification of the cells, and several subtypes emerged within that as well. And the proportions of cells that are in each of those clusters that came out of the unbiased clustering correlate with the age groups. So there are some of these clusters of cells. A lot of these ones are in the young dominant, then they have these clusters which become middle-aged dominant, and these clusters were classified as old dominant. And they went into looking at Violin plots of the expression levels of 14 of the microglial homeostasis-associated genes and inflammatory-related genes in each of those clusters. And we see some of them have pretty big upregulation in the age-dominant clusters compared to the young and middle. We then mapped out pseudotime trajectories, and this is something I hadn't looked at before, but I think it's kind of interesting. So this is taking the single cells and mapping the phenotype of young versus old, because you've got all these thousands of cells that are being profiled, and you can follow kind of the trajectory of the young profile cells through the old profile cells across, or the movement of kind of all of the cells start at the bottom here, and then as they move across like aging or the pattern of progression, they get more and more like these other phenotypes. And so it kind of gives you a temporal projection of cells kind of aging through that process. And then looking at the correlation of the ADAM genes with that pseudotemporal expression pattern, that correlates as well, where you get higher expression over time in the positively correlated genes and less over time with the negatively correlated genes. We then looked at ATAC-seq for accessibility of the chromatin sites and showed the accessibility increases with age around the transcript sites, and then showed some of the specifics here that came out, again showing that you end up with higher accessibility with age in some of these markers. And these are specifically in promoter regions for some of those genes that came up in the ADAM set. And then they showed also the kind of more detailed heat maps of the differentially accessible chromatin promoters or transcription factors for the ADAM genes, showing that you do indeed start to see changes that progress as you might expect with the gene changes, the expression changes, suggesting that their methylation is driving some of these expression changes that are shown. The next thing I wanted to look at is the response to LPS, so these being damage response type cells, so induced damage in C is do the microglial activations from LPS change over young versus old? And we do see the LPS versus PBS clusters separately, and then within the LPS or the PBS, you have pretty clear clusters of the responses being different by age group in each of those. And then again, the full clustering of by the unsupervised classification. And PCA of that, again, showing some distinct groupings of young versus old and LPS challenged. And again, diving into the expression level changes, we do see that there are many of these that start to change across cluster as well. And the annotations by genontology or IPA show differences in the immune response, interferon response like cells across those processes along this. Then they did these volcano plots of the response to LPS challenge, looking at LPS versus PBS in 3-month-old, LPS versus PBS in 14-month, and then LPS versus PBS in 24-month, and show that there are differences in the response profile that you get depending on which age group you're in. And with that, then looking into the genes related to cytokine production and phagocytosis, what showed is that there's a reduced response in older ages to the LPS. So the younger cells have a much higher upregulation of the genes related to cytokine production and phagocytosis compared to the old. And then looking at expression of specific markers of those, verifying the same thing, that they all have, the expected response to the, have a response to the LPS, but the aged have less of a response than the others, and some of these showing that the aged are higher just to begin with than the young are. Looked then at ATAC-seq for the chromatin accessibility again, and clustered those, and here showing the young versus with LPS versus PBS, you get this pattern. The old versus young with the PBS, and a similar pattern. Don't know what the specific points are, but the implication was that that old and LPS have some similarities in their methylation signature. And then looking at the LPS challenged, again, now with LPS of old versus young, and old versus middle-aged, and then middle-aged versus young. We get a few differential points that are accessible there. All right, the next section that they did, they established this new method of 3xDR microglia, and this is an interesting new model that they're proposing. They treat with a molecule that causes a depletion of the microglial population, and then wait for 20 days, and the microglia repopulate and grow, and then they hit them again to wipe out all of the microglia, and then they repopulate again. and then do it again. So they do this 3 times where they basically like kill off all the microglia and then let them repopulate. And when they get through this, they find that the resulting set of microglia have what they call an aged-like phenotype with reduced tumor length, transcriptional changes that are distinct from the controls, and a pattern matching, at least with looking at the ADAM genes that they had identified from aging, that you get an increase in the positively correlated ADAM genes and a decrease in the negatively correlated ADAM genes. So that they're purporting this to be a model of microglial aging where you effectively leave the rest of the cell types alone, but now have just aged microglia. Whether that is valid or not, it's a brand new model, so we can certainly have some conversation around that. Looking here at correlation of specific points, you do see a correlation of like old versus young and the 3XDR versus young in the response to LPS. Looking at the t-SNE plots, with the expression levels, you see changes in most of these. Interestingly, they said there are 545 differentially expressed genes with this model, and they only showed a handful of them, and they didn't really explain why. And so, that kind of makes me wonder about the rest of the conclusions drawn about the pathways and whatnot from this. But just, looking at the data, we do see clustering by age across these, and progression of pseudotime that are similar, at least looking at the single-cell microglia from the 3XDR and the aged. The 3XDR genes, there's very little overlap with the activation genes, which was similar to the naturally aged phenotype as well. So that at least, this is not just driving microglial activation, but seems to be more similar to the aging profile. The 3XDR microglia also have a distinct morphology where we see the cell body size is increased and the level of overlap of cells is decreased. So if you see the pictures here, you end up with these really big and disconnected microglia compared to the control. And then looking at the biological processes with IPA, similar things to what we saw with the aging and correlation of the ADAM genes and the damaged genes there. So, further supporting their stance that the 3XDR is a model of the aging. And then they looked at confocal imaging and showed upregulation of a couple of the points that they had of their ADAM genes of OPN and AXL. The next thing they went into was in vivo cognitive assessment to see if there were cognitive changes with the 3XDR model, and they showed that 3XDR leads to cognitive decline and myelination impairment in non-aged mice, reduced, and so here we have, this was a novel object recognition test where there's pre-training, put the mice together, day 1, they put 2 objects in, day 2, they have the same 2 objects, day 3, or stage 3, rather they replace one of the objects with the other. And in stage 1, there's very little response, and they're all just there. In stage 2, the older mice are looking at or spend more time exploring. In stage 3, the young mice, they're very excited to check out the novel object on the right side, and don't care about the object on the left side. The aged or the aged mice or the 3XDR don't really respond to the new novel object. The takeaway there. So suggesting that there is a decline in that recognition. They also did the 3XDR mice in the Y maze, and again showing that there's a deficit in the 3XDR in spatial learning, and then did the Morris water maze, and again showed that the 3XDR have a deficit in the spatial learning with the water maze. And then looking at myelination, they showed that 3XDR do have deficient myelination in the cortex. Okay, the next piece is single-cell RNA-seq to look at all of the cell types in the 3XDR, and they show, again, an age-like phenotype, and they show that there's some crosstalk with other cell types within the model. It's the same 3XDR model that was done. Looked at expression levels of cell type markers and show that they're able to identify the cell types based on their markers. And then looked at cell phone DB to measure ligand receptor interactions for cell-to-cell communication, connections between microglia and these other cell types, and show that there are some differences in control versus the 3XDR of the types of connections that are happening here between those. A lot of data buried in this cross type of oligodendrocytes and microglia and the types of connections, but punchline here being that there are differences in the way the cells are communicating with each other as far as ligand receptor interactions. Probably worth pausing and staring through that if you're interested in. And then also they use cell chat to do a similar thing, looking at molecule-molecule interactions between the cells, and again show a shift in the control versus 3XDR on many of these cell-to-cell communication methods.